Alright guys, second on the list, we're going to go through cellular respiration. I've tried to put it all together on one screen so we can view all of the different processes in one big picture. Now, I'll bring your attention to some of the key aspects here. So, originally we have glycolysis, where we're breaking down this glucose molecule, right? And see this 6-carbon glucose molecule. And the way we're going to do that is we're going to use an initial investment of energy, where we take ATP, we break it down to ADP, and we add these phosphate groups that you can see right here on the ends. Now, we add those phosphate groups on the end, and you'll probably notice, if you just watched the photosynthesis videos, you'll see that in the photosynthesis reactions, there happens to also be a time when we see a molecule with 6 carbons in it and 2 phosphate groups on the end. It's a different molecule, but in either case, remember, when you have a 6-carbon molecule with 2 phosphate groups on the end, it's unstable and it tends to break down. So just like that, we have the 6-carbon molecule, 2 phosphate groups on the end, it splits in half. When it splits in half, you create 2 3-carbon molecules with these phosphate groups on the end. Alright? So, now we're going to start the whole process of where we're using these electron carrier molecules, NAD plus and FAD, to steal hydrogens from these carbon-based molecules so that we can use them down here in the electron transport chain to power the electron transport chain. So, we've created these 2 3-carbon molecules with phosphate groups on the end. Then what's going to happen is you have some phosphate in your body from your diet, and these electron carrier molecules are going to come by, steal some hydrogens from this 3-carbon carbon-based molecule, and that stealing of the hydrogen is going to leave a blank spot on that molecule. Phosphate's going to come by and attach. So now we have 2 phosphates on the end of 2 of these 3-carbon molecules. So you consumed this glucose, right? Maybe you happen to consume it from a plant, right? Maybe it was stored in the form of starch, you broke it down to glucose, right? Maybe, in any case, it was made by a plant. So you consume this glucose, and you broke it down in half by making an unstable molecule. You've had an electron carrier molecule come by, steal a hydrogen, more phosphate groups attached. Now, we're going to reverse this whole process. So now we're going to steal these phosphate groups on the end of each one of these 3-carbon molecules. When we do that, when we steal a phosphate, steal a phosphate, steal a phosphate, steal a phosphate, we are going to create 4 ATP by ADP coming by and stealing off these phosphate groups. Then, we're left with 2 3-carbon molecules. Okay? So 2 3-carbon molecules are made, and we call those pyruvate. Now remember, pyruvate's going to go on to the transition reaction or the preparation for the Krebs cycle. Some key parts of this whole process are that these electron carrier molecules, NAD plus and FAD, come from B vitamins in your diet. Now don't confuse that with photosynthesis, where we talked about the pigments that are in the photosystems that can be chlorophyll or carotenes, and the most popular carotene we're all familiar with is beta carotene, because that is also known as vitamin A. However, these electron carrier molecules come from vitamin B, including this molecule up here called coenzyme A. So, you created these 2 3-carbon molecules, and you would have 2 of these side by side, right? But that's way too much to draw, so we just have 1 of them. So we have this pyruvate molecule come by, go and start the transition reaction, and all we're going to do in the transition reaction is change or rearrange these carbon atoms and rip off some of them, exhale them as CO2. Now, we have this pyruvate molecule. We're going to rip off one of those carbons and release it as CO2. Well, where the heck is that CO2 used? If you've been following me to this point, this might blow your mind. So, this CO2 right here comes from the CO2 that's produced in the preparation for the Krebs cycle and in the Krebs cycle. So where do we get the carbon dioxide that's used in the light independent reactions of photosynthesis? We get them from the preparation for and the Krebs cycle, where we release those carbon atoms as CO2. So we have pyruvate, we rip off a carbon, we create CO2, And we are then going to reuse another one of those electron carrier molecules. One's going to come by. It's going to steal a hydrogen atom from this acetyl group. So we had a 3-carbon molecule, happens to be called pyruvate, lose a carbon, exhale it as CO2. Now you have a molecule called acetyl, just a 2-carbon molecule. Then this electron carrier molecule is going to come by, steal a hydrogen, leaves a blank spot for this chunk of vitamin B. Vitamin B attaches to this 2-carbon molecule called acetyl. Now it's called acetyl-CoA. So far, we've made a couple NADHs that we're then going to eventually use down here in this electron transport chain, right? That's the whole point of all of these steps. Now, some key aspects before we enter the Krebs cycle in more details. Remember, this transition reaction right here is only entered if you have oxygen available. This oxygen, you see, is necessary to keep this whole assembly line going at the very end of this electron transport chain. And that's why it's necessary for you to breathe in oxygen. Now, if there wasn't any oxygen available, you would undergo anaerobic respiration or fermentation. Remember, for us, we produce lactic acid as the more advanced animals, but bacteria and some fungi do alcoholic fermentation. And the key process with alcoholic fermentation is you undergo glycolysis, and then fermentation occurs where you just keep recycling these NAD plus molecules over and over. But oxygen's available, you undergo the rest of the aerobic cellular respiration pathway. Now remember, depending on what kind of shape you're in, you have to run up to Atkins' library. You may not be in too great a shape, so sometimes you also do fermentation because your aerobic cellular respiration cannot keep up. And that's why you get lactic acid in your muscles, and that's where the soreness comes from that lactic acid fermentation. So, in any case, we've undergone the transition reaction. We have acetyl coenzyme A. It's going to enter into this Krebs cycle. Krebs cycle, pretty long and complicated, right? But remember, we just have this North Carolina, all for nothing to help you remember what comes in and out. But the Krebs cycle is similar to something you've seen before. We're simply taking a 2-carbon molecule with the coenzyme A and shoving it into a molecule that already exists inside of your bodies. Where does it exist from? You got it from nutrients from your mom when you first started this process when you were developing fetus. So, similar to the light independent / dark / synthesis / Calvin cycle reactions, we took a molecule and we shoved it into another molecule. And that's the same thing we're doing in the Krebs cycle over here. We're taking this acetyl coenzyme A molecule and we're shoving it into this 4-carbon molecule. You don't need to know the name of it, not important. You shove 2 carbons into a 4-carbon molecule. How does it do that, you say? It uses this special chunk of a B vitamin to attach itself to this 4-carbon molecule, and then it gets rid of that coenzyme A molecule. Now, 2 plus 4 equals this 6-carbon molecule down here. Then we just start rearranging that 6-carbon molecule, stealing hydrogens from it. NAD plus comes by, steals the hydrogen, and then we rip off the CO2, exhale it as CO2 to be used in the light-independent reactions of photosynthesis. We do that 2 times. Eventually we make this 4-carbon molecule that's really, really special. So this 4-carbon molecule is pretty neat. It's kind of like ATP synthase, where it can take ADP and connect it to phosphate to make this ATP molecule. It's not ATP synthase, it's just really cool because it's like it in that it can make ATP. So this 4-carbon molecule makes some ATP. Then we steal some more hydrogens from it. This is the first time we see this FAD electron carrier molecule that we'll later use in the electron transport chain. And we're going to steal a couple hydrogens from this 4-carbon molecule. Then, at the very end, we're going to recreate this 4-carbon molecule we start this whole process with by stealing another hydrogen from it. So remember, we had 2 pyruvates originally, right? And these 2 pyruvates are going to come over here into this transition reaction, which is why we say it happens twice, because we have 2 of these molecules we need to take care of. So although we only make what you see to be 1, 2, 3 molecules of NADH in the Krebs cycle, we actually make 6, right? Because this thing happens twice due to these 2 original pyruvate molecules when oxygen is available. So that's what happens in the Krebs cycle between each of these steps. Now remember, the whole point of all of these things, originally starting in the cytoplasm, moving to the matrix of the mitochondria, and remember, here's your mitochondria right here. The whole point of all of these was so that we can power the electron transport chain. So we power these electron transport chains by dropping off hydrogens and electrons by these electron carrier molecules. So remember, we have this NADH that we made in glycolysis. We have some NADH we made in the preparation for the Krebs cycle. We have a bunch of NADHs that we made in the Krebs cycle, all coming to this electron transport chain and dropping off electrons and hydrogens to power it. We also have this FADH2 molecule coming by and dropping off electrons and hydrogens. So here, we're looking at the electron transport chain. Now, you won't need to draw the transition reaction or the Krebs cycle or the glycolysis in detail, but if you have an intimate understanding of how all of these steps work, then you understand what goes in and what comes out. These inputs and outputs are also on your lecture guide as well. So, we have these electron carrier molecules at the electron transport chain, dropping off these electrons, dropping off these hydrogens, and they're going to power this electron transport chain. So they drop off these electrons, which shove their way through these proteins. These proteins act like vacuums, and they suck these hydrogens inside of the inner membrane space. So that's inside of here, right? So we are sucking these hydrogens in. Remember, we're in the wall right here of the mitochondrial matrix, and you can see this is what's blown up right here. So these hydrogen ions that you can see being pumped into the inner membrane space essentially are being pumped to fill up this outside bag right here of the mitochondrial matrix. You can see the matrix is what's inside of this inner bag. Okay? So we have these electron carrier molecules dropping off electrons, shoving their way through these proteins, and pumping these hydrogens across these proteins as well. Now we know we get a ton of hydrogens on the inside, just like in photosynthesis. Things in biology want to move from high concentration to low concentration. These hydrogen ions shove their way through this ATP synthase molecule, powers that ATP synthase, causes it to rotate. That rotation causes that ADP to grab phosphates and connect it to create ATP. And that's the way we make ATP, actually 32 from the process of the electron transport chain. Remember, to keep this whole process going, you need something to accept these electrons. That's why oxygen is called the terminal electron acceptor. Okay? That's really awesome looking. Terminal electron acceptor, TEE. So, also, super important, this terminal electron acceptor, oxygen, is also binded or bound to hydrogen ions and then eventually excreted from the body, and then you pee it out. If you happen to go pee outside, maybe you start this whole process of photosynthesis over where you're splitting these water molecules, making this oxygen that we then need to power that electron transport chain. Most of the time you're probably not peeing outside, but a lot of animals are. It evaporates, goes into the rain, rains down, provides water for the water splitting photosystem. Byproduct is oxygen. We breathe that oxygen in, and we need it to power this electron transport chain at the very end of aerobic cellular respiration. Okay? So that's that process in a nutshell. Make sure you go over this, maybe watch it a couple times. May help you understand how all of these things are connected, what goes in, what comes out. Be super familiar with the electron transport chain.